thank you to the organizers. I'll be talking about our work on fully unsupervised reconstruction of three-dimensional protein structures from two-dimensional cryo-EM images. And specifically, we'd like to reconstruct heterogeneous dynamical protein structures. So as the main biological motivation for this work, as we all know, large macromolecular complexes underlie all essential biological processes. And of course, we'd like to obtain the structures of these complexes in order to understand how they work. And as a first pass, it'd be nice to obtain a static structure, but what we'd really like to know is how they move in relation to how they function. And so in pursuit of this goal, we can use cryo-EM, which stands for cryo and electron microscopy. Over the last decade or so, there have been dramatic advances in this technology, mostly driven by better hardware, so these direct electron detectors, and by better image processing algorithms, so these probabilistic methods that are typically implemented on GPUs. And so just as a quick background of cryo-EM image formation, so in a cryo-EM imaging experiment, you have a purified sample of your target molecule of interest, whose structure you want to determine. This is frozen in a very thin layer of ice, and it's imaged with an electron beam, and your recorded image is typically modeled as the integral of this particle's three-dimensional electron density function along the imaging axis. And so these molecules are randomly oriented as you flash freeze them. So there's an orientation, unknown orientation in each of these particles. And they're also, unlike the schematic, extremely noisy due to the low electron dose required to limit radiation damage. And so that's the forward model at a very high level. The task of reconstruction is to solve the inverse problem of going from images, so a typical number being in the hundreds of thousands, to the 3D electron density. So here's a real data set of the ADS ribosome. So a couple of challenges, as I mentioned, they're the unknown particle poses, so you don't know the orientation of each of these volumes within the image. There's a low signal-to-noise ratio. You have to take into account image-degrading filters in microscopy, such as the point spread function of the microscope, discretization of measurements, but then the problem that we're particularly interested in is the so-called heterogeneity problem. So this is where you break the standard assumption that each of these structures and that each of these volumes within each image is the exact same static structure. But before I talk about heterogeneous reconstruction, just one more quick piece of background for the homogeneous case. So cryo-EM, like traditional cryo-EM algorithms, all use what's called the Fourier slice theorem to relate these two-dimensional projection images to the three-dimensional volumes. And so the Fourier slice theorem states that the Fourier transform of these 2D projection images is a central slice out of the three-dimensional Fourier transform of the volume, perpendicular to the projection direction. And so this suggests a method for reconstruction. Given a bunch of images, if we knew their orientations, we could just take their Fourier transforms, insert these slices at their particular orientation in the three-dimensional Fourier transform, build up this three-dimensional Fourier transform, and then take the inverse FFT to get your real space volume. And so this works well in the homogeneous case. Traditional reconstruction algorithms use the Fourier slice theorem. However, the so-called heterogeneity problem, I think, is actually a major opportunity in cryo-EM. So this is definitely like throws a wrench in the reconstruction task. Each image contains a unique molecule, but because we actually capture the data for these heterogeneous states, we can actually interrogate the diverse functional states of these different proteins. So standard approaches for heterogeneous reconstruction, other than just discarding your heterogeneous data, is something called multi-class reconstruction, which models a discrete mixture model of k independent volumes. You don't know the nature or the number of states a priori. For example, your proteins might lie along a continuum of states. And so this, in general, the identification and analysis of heterogeneity, especially its continuous forms, is an open problem for cryo-EM. And so what we'd like to do is learn a continuous low-dimensional manifold of structures in a fully unsupervised fashion. So our particular method is called CryoDragon, which stands for Deep Reconstructing Generative Networks. We develop, in brief, coordinate-based neural networks to directly approximate the three-dimensional structure. We use a Fourier space image encoder volume decoder architecture based on the VAE to learn a latent variable describing the protein's structural heterogeneity. We also employ an exact inference for the pose variables and variational inference for the heterogeneity. So our coordinate-based neural networks can be best understood in contrast to traditional algorithms. So instead of representing our volume, our structure, as discrete points in a three-dimensional voxel array, we instead use a neural network to approximate the continuous function, which maps from 3D Cartesian coordinates to the electron density at that particular location in three-dimensional space. Instead of directly feeding in the three-dimensional coordinates, we use a fixed positional encoding function to featureize the coordinates in terms of sinusoids of varying frequency. And then the reason why we would do this is to model heterogeneity. So in this traditional multi-class reconstruction framework, we would model a mixture of k independent separate voxel arrays, whereas in our approach, we can simply extend the input dimension of our neural network to include a latent variable z denoting heterogeneity. So that becomes a generative model in our framework. That's the volume decoder. This is a schematic of the overall architecture. We have an image encoder, which takes an input image and produces a distribution in latent space describing this particular image's heterogeneity. And then we know that each image by the image formation model is a slice out of the three-dimensional volume. So we can take those three-dimensional coordinates and that sample from that approximate posterior, feed that into the volume decoder, and reconstruct the image pixel by pixel. We don't know the orientation of the slice a priori, so instead we do this search over all possible, or over discretization of all possible orientations and in-plane shifts using a branch and bound global optimization procedure. And so I also want to note that this coordinate-based representation is really nice. It enforces geometric consistency between these two-dimensional views. Each of these two-dimensional projections should share a common line, line in common. So this is all enforced by our positional-based volume decoder. And so we want to evaluate this first on synthetic data. So in the simplest case, we create this heterogeneous complex, heterogeneous data set containing a model protein complex with 1 degree of freedom. And so we have this atomic model here, and I rotate one of the dihedral angles connecting this blue domain of the protein to the rest of the complex. I sample 100 atomic models along this reaction coordinate and generate 500 randomly oriented and translated projections for each model, yielding a total data set of 50,000 images. And so the question for a method is, can we reconstruct this entire continuum of states? And so if we train a one-dimensional latent variable model, here I'm showing the predicted latent code for each image produced by the encoder along the true reaction coordinate, which we know in this case because it's synthetic data, and we can see that it correlates very nicely. We can also sample or generate the volumes corresponding to the different points in the latent code, and so here are 10 different structures shown at high isosurface and low isosurface representation for the volume. You can see that we reconstruct the ground truth structure. We can generate movies, which look kind of silly in this very synthetic data set. And in contrast, CryoSpark, which is an existing tool for heterogeneous or for traditional cryo reconstruction, its multi-class reconstruction is with 3 classes is only able to reconstruct 1, maybe 2 volumes that resemble the ground truth, with a third having very weird alignment artifacts. And in general, you also lose the underlying heterogeneity in the data. And so there are a couple of additional data sets we've generated that have more complex latent structure. In this linear 2D data set, we have 2 independent domains of the protein complex that are allowed to move independently. In the circular 1D data set, we have 1 domain of the protein which rotates 360 degrees. We have a discrete mixture of 10 different classes. And what I'm showing here is the data set embedded in latent space. And what's really cool is that the topology of our data distribution in latent space matches the topology of the underlying heterogeneity. And we also quantify the accuracy of our volumes using an FSC resolution metric, which is a standard in the field. And then finally, I just wanted to show some results on some real cryo reconstructions. So this is a data set containing a mixture of ribosome assembly intermediates. The biological question was trying to understand how the ribosome is formed, and this is my advisor Joey's postdoctoral work. And so through many manual iterations of this multi-class reconstruction, they identified 4 major classes of the large ribosomal subunit from this multi-class reconstruction approach. And here what I'm showing is our method training a 10-dimensional latent variable model. And this is a distribution of data in latent space colored by the class assignments from the publication. And so you can see that the 4 clusters for the major classes come out really nicely. And here are the structures generated at these points at the cluster centers, and the resolutions of these structures match the resolution of the publication. We can also sample additional volumes from the latent space. This blue cluster all the way on the right actually corresponds to an impurity during the sample preparation, which is a 70S ribosome, and so this comes out automatically in our reconstruction method. And here are just 4 additional structures sampled from this particular class, this particular purple cluster, showing the heterogeneity which corresponds to the known intermediate states for the ribosome during assembly. And then finally, I just want to have one last data set of the precatalytic spliceosome. So this structure of the spliceosome was the first structure published in 2017. And you can see that there's a low-resolution core, or high-resolution core with low-resolution distal elements, and the structure was actually resolved separately, or the different domains of the structure were resolved separately, and then pieced together just to form this composite map because there was so much heterogeneity in this data set. And so, in contrast, this is the reconstruction that we get using our method. This is just one particular latent traversal in latent space, but we can see that there's this stable core and lots of motion in the distal elements of the complex. And what we can do is not just do one latent traversal, we can do a random walk along latent space. So this is 132 sampled structures. We do this random walk and we can see complex heterogeneity at the domain interfaces. And so in contrast to the traditional approaches, which are able not even to get one structure, we can sample this entire conformational ensemble that really, to me, resembles a electrodynamics trajectory, almost. And so in conclusion, this is our method for fully unsupervised reconstruction of a continuous low-dimensional random folder protein structures from cryo-EM data. It's the first method to apply neural networks to the cryo-EM reconstruction task, and the first that can learn a continually heterogeneous representation of protein structure from cryo-EM data. And we started sharing this method to different research groups who were unable to use the traditional algorithms for reconstruction of their protein structures, and we hope to identify new structures from real cryo-EM data. So with that, I'd like to thank both my labs as well as other people who have provided good feedback, and thank you. Differential, what, pardon?
Like ray tracing for images, right? Where you have a volume, but then you generate it through the image and can differentiate a bit. Have you thought about any thing like that for encoders so you can model kind of the projection correctly?
So the image formation model is slightly different. The images are actually these projection images, so like integrals through the density along the imaging axis, whereas in other like domains of 3D shape reconstruction and computer vision, they're like surfaces where you have this ray tracing. So I'm not exactly sure how that, like we already directly model the image formation process by treating our images in Fourier space as slices of the volume. Yeah.
That's really cool. I guess I have two sort of unrelated questions. One of them just has to do with that ribosome example, and the red cluster seems to actually be in two clusters if you look at that. And the other one is
Yes.
these kind of methods obviously benefit from lots of data. How easy have you found it to, we'd like it in principle just to scale up and automatically slurp in data that's all that is out there. Do you have ways to do that in this domain?
Yeah, so I guess like answering the second question first, one thing about this method is that we train a separate model per experimental data set. So for each new, I guess, cryo-EM data set, we're training a separate model, and we don't really expect it to generalize to unseen protein structures or proteins that aren't in the particular data set. Although it is kind of an interesting question of can we take advantage of all the structures, all the protein structures that we know now through cryo-EM. And then to answer your first question, I think, so the answer is yes. The actual published data set had 13 different structures of the ribosome. So within these 4 major classes, you have minor variation in classes C, D, and E. And then if you're particularly curious, oh wait, it's not in here. But the different clusters within the different clusters within this red cluster, the red major cluster, correspond to these minor states of ribosome assembly, because we know it's this complex parallel process. Yeah. Yeah.
If I understood correctly, you need to choose the dimensionality of Z before you apply.
Correct.
Right?
Yeah.
Is there like guidance of how to choose that or data? And also, how does it change for different dimensionalities?
Yeah, so in all the examples that I've shown here, other than that first synthetic example with the one-dimensional motion, they're all trained with 10-dimensional latent variables, and that's just a hyperparameter that you have to select in your architecture before you train. You could cross-validate the latent dimension, but so far I haven't done that, and what we've seen is that as long as the dimensionality of your latent variable exceeds the dimensionality of the heterogeneity you're trying to model, you're so far okay. But that's definitely something that maybe could be tuned. So that would be the next step. So after like the output of cryo-EM reconstruction is this electron density map, but then as a practitioner or as a biologist, you want the atomic coordinates. So that's like the next step in the cryo-EM pipeline, and maybe there's some ways of incorporating the known like sequence information in how we model the structures. Right now, our volume representation is just a MLP, so it's a very like naive representation for the volume, which is very flexible, but maybe we can do something better by incorporating the atomic positions in some way. Yeah. Cool, thank you.